Okay, so my topic was the coevolution of plants and their pollinators. This presentation more specifically focuses on the fine-tuned bee flower coevolutionary state hidden within multiple pollination interactions. So let's begin with pollination. Pollination is the most common form of mutualism between plants and animals. Angiosperms, which are plants that have flowers and produce seeds enclosed within a carpel, produce pollen to attract visitors, and in return, these visitors transport pollen and affect pollination. These plant pollinator interactions are predicted to be the products of coevolution between plants and insects. So, strict one to one coevolution is usually found between a plant and its insect partner, like a fig, wasp, and fig plants. But despite this, many angiosperms can be visited by pollinators that visit a multitude of flowering plant species. For generalist plants, the optimal phenotype expected would be an intermediate between all the pollinators' phenotypes. Um, so, because of this, a fine tuned specialist in mutualistic interaction between multiple flowers and multiple pollinators is not what they expected to see. So, the purpose of this experiment was to describe a fine tuned morphological specialization between an andrenate bee, Andrena lanicerae, and an early spring flower, Lanicera gracilipes, that is visited by multiple pollinators. So, they also show that the bee is finely adjusted to the morphology of the flower. And lastly, they discussed the implication of finely tuned um, one-to-one mutualistic state in the context of coevolution of pollination interactions between the bee and the flowering plant. So they investigated the pollination activities of the bees belonging to the species A. lanicerae, visiting the flowers of L. gracilipes, and A. lanicerae is oligolectic, meaning the bees collect pollen from a limited number of phylogenetically related species, and is mostly found on L. gracilipes. This flower blooms from March to May, and because it blooms very early, um, this flower is frequented by many different insects. So pollination experiments showed that the other visitors proved to be as effective pollinator of this plant as A. lanicerae. But despite this, these other visitors were only able to collect pollen. The nectar of this flower is almost exclusively collected by A. lanicerae. This is because the plant has a long, narrow corolla tube. Despite belonging to a group of short-tongued bees, A. lanicerae has extremely elongate distal part of their tongue, and pollinators with a short tongue are unable to reach the nectar. So, A. lanicerae females visit flowers of L. gracilipes to collect nectar, which is their own energy source, and to collect pollen, which is the energy source for larvae. The bee foraging activities indicate that when pollen was a primary goal, they sought pollen before they collected nectar. However, when nectar was a primary goal, they rarely looked for pollen. There are two possible reasons for this. The pollen has already been removed, or bees are in the pre nesting stage, so there was no requirement for pollen collection. The asymmetry in behavioral sequence may be explained by differences in resource availability between the pollen and the nectar. So, although a single flower has five anthers or pollen sacs, pollen is limited non-renewable resource. Pollen is usually removed by bees in the first couple days of bloom, and many flowers in a blooming period have very little pollen, but when it is available, bees collect it first. Pollen is so limited that many bees will sometimes tear off unopened flower buds to collect pollen, nectar, or both. Nectar is an extremely limited but renewable resource. During the flowering period for each blossom, the amount of nectar secreted daily is decreased rapidly from day 1 to day 6. Because the amount of nectar is always low and many flowers have no nectar, the bees must visit flowers frequently to collect nectar even if pollen is unavailable. The high frequency of these flower visits of A. lanicerae for nectar suggests that L. gracilipes provides nectar almost exclusively to this bee. So these graphs talk about what I just stated. So the first graph, graph A, shows the flower visits, and as you can see, for nectar, um, they visited the flower 850 times out of a total 1,171. Graph B shows the decrease in the nectar volume as the days progress. Graph C shows the amount of nectar found in open flowers, including all flowers. So this one shows all the flowers on the left, and then this on the right shows um, all the flowers, excluding the 23 that have absolutely no nectar. This graph here shows open pollination compared to control, which is just unopened buds. And as you can see, the open pollination ones have um, pretty much no pollen in them. Um, Morphology and function. So the head and mouth parts of three different bees were compared. So they compared lanicerae to its closely related A. halictoides and a distantly related A. hebes. Um, A. hebes is the second most common visitor to the flower L. gracipalis, but only for pollen. In contrast, A. halictoides, mostly allopatric to A. lanicerae, is closely related species that utilizes the funnel flower of W. hortensis. So within these three species, the female head is extremely elongate in A. lanicerae, which is this one here, and the length and width proportions of the head is significantly different among them. So this A. lanicerae, this head is 1.1 times as long as broad um, for the females as for this one, and then for A. halictoides, the female head is 1 times as long as broad, and then for A. hebes, it's 0.92 times as long as broad. So they also found that the oculomalar space of the head is also considerably elongate in A. lanicerae and A. halictoides compared with the more typical short malar space of A. hebes. So if you look here, this is the ocular malar space for A. lanicerae and A. halictoides, and if you look here, it's much shorter on A. hebes. Um, the proportion of the malar space to the head width is also significantly different among the species at 0.12 millimeters, 0.09 millimeters, and zero point zero 0.04 millimeters in A. lanicerae, A. halictoides, and A. hebes, respectively. This suggests that the elongation of the head in A. lanicerae is an adaptation to tube-shaped flowers. Um, the mouth parts for both A. lanicerae and A. halictoides females also exhibit distinct specializations. So the distal part of the tongue is greatly extended in A. lanicerae. So if you look here, resembling a string and fairly elongated in A. halictoides, which is this one here, compared to that one which of A. hebes. So it should be noted that among approximately 1,500 species of the genus Andrena, A. lanicerae and A. halictoides are the only two species possessing an elongate distal portion of the glossa, hence their placement in the subgenus Stenolomelissa. The paraglossa covering the basal part is rudimentary in both A. lanicerae and A. halictoides. So in both A. lanicerae and A. halictoides, there is also a distinctive modification in the maxillary glia covering the basal proboscis dorsally. So this is what I'm talking about here. And the brush-like structure called the galeal comb cleaning apparatus that is located on the face of the glia, shared with almost all short-tongued bees, undergone reduction to 1 to 3 bristles, the loss of almost all setae in the glial comb, and the considerable reduction in the size of the paraglossa are unique features found elsewhere in the long-tongued bees, but are atypical of short-tongued bees, indicating convergence between long-tongued and short long-tongued bees, and A. Lanisterei and A. halitoides. So here's where I was talking about. So the measurements of A. lanisterei correspond directly to the tube-like flower of L. gracilipes. Because nectar quantities are usually small and located at the base of the funnel shaped flower, the bee has to fully extend its proboscis to collect it. They found agreement between the effective length of the bees and proboscis and that of corolla tube in both the Hino and Kyoto populations. So the nectar reaching rate of all bees in the Hino population is 100% in that all bees have access to the nectar. And then in the Kyoto population, it's 91%. And then the departure index for the Hino and Kyoto population are 0.76 millimeters and 0.6, I'm sorry, 0 millimeters respectively. And these two measures, you can see that down here, and these two measures indicate that there is a close match between the proboscis length of the corolla tube in both of these populations. So these bees place their head deep in the flower when it's searching for nectar. However, when collecting pollen from the anthers, it hangs on the flower without inserting its head into the corolla. When they retrieve the nectar, the bee extends its mouth parts within the corolla tube, and the proboscis, which is highly flexible, fully extends when necessary. So the results demonstrate a one-to-one morphological match between the proboscis and the corolla tube. Both morphological and behavioral adaptation of the bee, as well as nectar production by the flower, show that this flower pollinator interaction is highly mutualistic. The mutualism is supported by the close matching between the effective length of the head and mouth parts of the bee and the corolla tube in two separate populations. So flowering plants are typically visited by many pollinator species and pollinators often visit many different flower species. This case an example of both pollinator and flower being generalists. A. lanisterei often solely visits the flower L. gracilipes, exhibiting narrow oligolecty. L. gracilipes appears as a generalist because it is visited by many flowers. Hence, this relationship shows an interaction between an oligolectic bee and a generalist flower. The matching of the head and mouth parts of the bee to the tube length of the flower indicates that both the flower and the bee might have co-evolved. So the data shows that pollination effectiveness of one visit by other frequently visiting bees is almost equivalent to A. Lanisterei. Assuming that other bees constitute evolutionary partners, the nectar of L. gracilipes should be accessible to these other bees. However, despite them being effective pollinators, they aren't able to reach the nectar in the flower. So this indicates that these other bees are simply riders or freeloaders who abandon the patch after a single visit for pollen. Close examination of these interactions between A. lanisterei and L. gracilipes reveals a tight one-to-one relationship. Among all visitors, L. gracilipes is the most frequent and essentially the only one to collect both nectar and pollen. Coevolution is often discussed in the context of specialists and generalists, where both pollinators and flower specialists have evolved from generalists and vice versa. This is why we cannot identify mutual partners so easily. Reason being, there are often so many so-called rider species that utilize or steal pollen nectar from the flower. As with L. gracilipes, many apparently generalist flowers may be in fact finely tuned to a single pollinator. The oligolectic pollen diet of Adrena bees also constitutes evidence for also constitutes evidence for this narrow specialization. In almost all symbiotic and mutualistic systems, we may find species-specific one-to-one interactions. So in conclusion, with respect to pollination ecology, their findings imply that adaptation or coevolution and mutualist relationships or interactions are two different things. Multiple mutual interactions do not mean Multiple coadaptations. Many angiosperms may have target pollinator that guarantees the pollination of their own flowers. Insect pollinators with only a short active period may have a target plant species when they search for flowers, as does a Linicerae. Hence, one-to-one cohabitation might have evolved and been retained within multiple pollination interactions. Any questions?
Done, Taylor. That was seemed like an impressive amount of work, and you had to digest and put out there for us.
Yeah.
Um, what attracted you to this talk or this idea?
So, as I was looking for things to talk about, I came across one paper that was talking about how Darwin had discovered, the idea of coevolution before he knew what coevolution was. And so I just kind of ran with that and found this paper that I liked. So, yeah.
Are you a grad student? Because you
Yes.
seem, have you, what year or what semester are you in it right now?
This is my second year.
Okay. Are you working with someone right now?
Not right now.
Okay. Well, you look like you had some really good stuff. I don't know if you want to do some, if I don't know if we have time, if you're interested in some more project kind of stuff, you might want to visit with me, but
Okay.
you really seemed like you're really into entomology, which is pretty cool. And I don't know if you want to put it in participant mode, then we can see if anybody else has any questions.
Sorry.
And there's a really good, lots of information.
Thank you.
Are you trying to graduate this semester though?
No.
When are you trying to graduate?
Maybe next semester, so in the fall of this year.
Okay. Anybody have any questions for Taylor or suggestions or comments?
What was the region of that study?
It was conducted in Japan, so like around Tokyo is what I read on here.